point last time where we described the position of the upper motor neurons in the precentral gyrus and how those neurons sent a process out in the internal capsule, crossed the midline in the decusation of the pyramids, and descended the cord in the lateral corticospinal tract. We'll pick that up today where we're going to describe how those lower motor neurons synapse. We're going to pick that up today when we describe how those upper motor neurons synapse onto lower motor neurons in the ventral horn. And then we'll see that those lower motor neurons will exit the spinal cord as motor efferents and innervate muscle fibers in the periphery. And that will then give the idea of the motor unit. Remember, we described the motor unit last time as the lower motor neuron and the group of muscle fibers they innervate. So what we're going to do here now then is lay out the neuromuscular synapse. We're going to look at the synapse between the lower motor neuron and the muscle fiber, and we're going to find that the neurotransmitter that's being released is acetylcholine. And this acetylcholine then binds to nicotinic acetylcholine receptors. They will bind to nicotinic acetylcholine receptors on the muscle fiber. Those nicotinic acetylcholine receptors, when bound to acetylcholine, will then open and generate an excitatory postsynaptic potential. This story is the same as we've seen before. Once the EPSP is generated, that will open voltage-gated sodium channels, and the action potential now will propagate along the muscle fiber and down into a region called the T-tubules. So after we have delivered the action potential to the after we've delivered the neurotransmitter to the muscle fiber membrane, we'll then propagate the action potential along the sarcolemma and down into the T-tubule system, where the T-tubule will now meet the sarcoplasmic reticulum. So we'll see here that there's a set of membranes, the sarcolemma, the transverse tubule, and the sarcoplasmic reticulum. The sarcoplasmic reticulum is an intracellular bag of calcium. It's nothing more than an elaboration of the endoplasmic reticulum. And we'll see there now that at the point where the T-tubule membrane comes very, very close but does not join the sarcoplasmic reticulum membrane, at that point we have the gap between the two membranes bridged by proteins, the dihydropyridine receptors and the ryanodine receptors. The dihydropyridine receptors are transmembrane proteins in the T-tubule membrane. The ryanodine receptors are transmembrane proteins in the sarcoplasmic reticulum. And what we'll find is that the dihydropyridine receptor is also a voltage sensor. And when the action potential is delivered to this narrow gap between the T-tubule membrane and the sarcoplasmic reticulum, at that point, then that depolarization will move the cytoplasmic portion of the dihydropyridine receptor, which had been stuck in the channel of the ryanodine receptor. When we unstick that channel, it opens the ryanodine receptor in such a way that calcium can now pour out from the sarcoplasmic reticulum and into the cytoplasm. That brings us to this interface between the membrane system and the cytoskeleton. The cytoskeleton in skeletal muscle is organized in a repeating pattern called sarcomeres. Every micrometer or two, there's a sarcomere. And that sarcomere is bounded by a structure called a Z-line, or alpha-actinin. In between the Z-lines, there will be thin filaments, actin thin filaments, which are composed of actin, troponin C, and tropomyosin. Troponin C is a calcium binding protein. So when calcium binds to troponin C, it causes the tropomyosin to move. When the tropomyosin moves, the myosin binding site on actin is exposed. The myosin head then reaches up, contacts that actin thin filament, forms a bridge. And once the bridge is formed, then ADP and PI leave, the myosin head moves, and then when ATP binds to it, it lets go and recocks. And we'll walk our way through that system. In the end, the entire process is brought to a close when ATP binds to the myosin head to make it let go of the actin thin filament, and then calcium is pumped back into the sarcoplasmic reticulum. So contraction N when calcium is removed from the cytoplasm and back into the sarcoplasmic reticulum. But where I want to start here is to look at a cross-section of a spinal cord. Out here at the side is the lateral corticospinal tract. And in the lateral corticospinal tract, we see the conductile regions. I'm going to show these as green dots. These are the conductile regions of the upper motor neurons. And these upper motor neurons are located in the precentral gyrus. And the precentral gyrus contains the motor cortex. So those upper motor neurons, the cell bodies of the upper motor neurons, reside in the precentral gyrus in the motor cortex. They send processes out, and at the appropriate place, these now will leave the lateral corticospinal tract and enter the ventral horn. In the ventral horn, they will synapse onto the input region of a lower motor neuron. And there will be hundreds of these lower motor neurons in the ventral horn. I'm just going to show 3 of them here. And as we've seen, those lower motor neurons whose cell bodies reside in the ventral or anterior horn exit the central nervous system. They exit the central nervous system in this structure known as the ventral or anterior root. Now, if we were going to follow all of these axons, we're going to follow the process of the red neuron out into the periphery. And we're going to follow the process of the purple neuron out into the periphery. And we're going to follow the process of the brown motor neuron into the periphery. And over here then, I'm going to lay out a series of muscle fibers. Each of these parallel lines is meant to represent a muscle fiber. So if we follow the path of the purple lower motor neuron, it comes on up, finds its way to the muscle, and it innervates. Let me number these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and 15. Now you remember the definition of a motor unit. A motor unit is the lower motor neuron and the group of muscle fibers. It innervates. So if we follow purple, and we arbitrarily say now the purple is going to form a synapse onto 2, 6, 9, and 11, then the motor unit is the lower motor neuron we've labeled purple, along with muscle fibers 2, 6, 10, and 11. We can repeat that pattern where red comes out and innervates 1, 3, 8, and 9. And brown now innervates everybody else. So 4, 5, 7, 12, 13, 14, and 15. So what does this mean? It means when the lower motor neuron is activated, when the lower motor neuron is activated by the upper motor neuron, when the lower motor neuron is activated by the upper motor neuron and fires action potentials, then lower motor neuron red is going to activate muscle fibers 1, 3, 8, and 9. Lower motor neuron purple is going to activate 2, 6, 10, and 11. So every time we activate a lower motor neuron, we now activate a group of muscle fibers. The other important point to be shown here is that each muscle fiber receives a synapse or receives input from only one lower motor neuron. So that means now action potentials in purple cause the contraction of 2, 6, 10, and 11, always and almost completely simultaneously. Similarly, brown now activates 4, 5, 7, and the 12, 13, 14, 15. So it's a larger motor unit. Now in biology, we find enormous variability in the size of motor units. The muscles of our back and our leg, the postural muscles, in those cases, a motor unit may be a single lower motor neuron and as many as a few hundred muscle fibers. Among the finer muscle movements that we control, such as our hand and the extraocular eye muscles, which move our eyeballs, in those cases, the motor unit may be a single lower motor neuron and no more than maybe 15 or 20 muscle fibers. So those motor units, which contain a single lower motor neuron and a small number of muscle fibers, those can control very delicate movements. So when we want delicate movements, we call up the motor units which contain a single lower motor neuron and a relatively small number of muscle fibers. When we want to get a stronger response, then what we'll do is we'll call up larger motor units, and when we want to cause a really strong muscle contraction, we'll then activate all of the motor neurons pretty much at the same time. And that's the reason why, if you imagine walking over to the countertop and using your hand to pick up a dime, it's a fairly delicate movement. That's done by the recruitment of a small number of lower motor neurons innervating fairly small motor units. But then the question arises, how do we get enough hand strength to pick up a bucket of cement? That is done by recruiting more and more and more motor units until all of the muscles in the hand are contracting almost simultaneously to generate enough strength to pick up the bucket of cement. But we can regulate the number of lower motor neurons that are active over a broad range by the number of upper motor neurons we recruit. So any questions to there? That's the concept of the motor unit. So now what I want to do is I want to drill down and think about what's happening at the interface between the lower motor neuron and the muscle fiber. So this is a typical muscle cell. So the typical muscle cell, a skeletal muscle cell, is large. In some cases, these muscle cells may be more than 1 centimeter in length. They may be as much as 50 to 100 micrometers in width. Now, these muscle cells are generated from the fusion of myoblasts. So that during development, we start out with individual single nucleated cells. And then as the muscle develops, these myoblasts come close together and they actually fuse one with the other. Each one of these myoblasts contributes a nucleus. So when we generate a muscle cell during development, we may have a few hundred of these individual myoblasts fusing together, each one contributing a nucleus. So these muscle cells are said to be multinucleate. These are gigantic cells which contain a few hundred or a few thousand nuclei that are all the result of the fusion of these myoblasts during development. But these are big cells. And as we implied from that image above, there's a single site along the muscle fiber membrane where the lower motor neuron forms a synapse. So this is where the lower motor neuron this is the lower motor neuron synapsing onto the muscle fiber. This is said to be the neuromuscular synapse. Now, the rules for the neuromuscular synapse are identical to the ones we've described previously, in that this output region of the lower motor neuron in thin section in the electron microscope is filled with synaptic vesicles. Some of those synaptic vesicles are plastered right up against the membrane. The vesicles are docked at sites called the active zone. And just as we described before, the hallmark of the active zone are these red voltage-gated calcium channels. Each one of the vesicles contains about 5,000 to 10,000 acetylcholine molecules. So from here, the pattern's the same. The action potential is initiated in the upper panel in the input region, or the action potential is initiated at the trigger zone of the lower motor neuron. Those action potentials propagate down to the output region. And that each one of those synaptic sites on those multiple muscle fibers, almost simultaneously, there's calcium influx, which leads to the fusion through the snare event of the plasma membrane and the synaptic vesicle membrane. When that happens, neurotransmitter is released. So this is acetylcholine. And again, we see the same pattern as we saw in the central nervous system. Here in the muscle fiber, let me put them up here. Here in the muscle fiber, there are going to be these nicotinic acetylcholine receptors. These are the very same receptors that Langley observed when he used his camel's hair brush and he marched along the muscle fiber and he reached the site where the lower motor neuron met the muscle fiber and the muscle contracted. The reason the muscle contracted was because the nicotine bound to these nicotinic acetylcholine receptors. And the nicotinic acetylcholine receptors are ligand-gated, ionotropic channels. They're cation channels that permit influx of sodium ions. So the pattern here is the same. We release the neurotransmitter now diffuses across the narrow gap called the synaptic cleft and binds to the nicotinic acetylcholine receptors. When the acetylcholine binds to the nicotinic acetylcholine receptors, if we were to put an electrode in here, we would see that the opening of the nicotinic acetylcholine receptors causes an excitatory postsynaptic potential. If we were to record this excitatory postsynaptic potential, we would see the membrane potential goes from about minus 70 millivolts all the way up to about minus 20 millivolts. What's really happening here is that when the action potential arrives at the output region of the lower motor neuron, almost simultaneously, something like 50 to maybe as many as 200 of these synaptic vesicles fuse the plasma membrane and release their contents. Each one of these opens several hundred, maybe up to maybe a couple thousand nicotinic acetylcholine receptors, each one of those contributing a little bit of influx of sodium ion so that we now get a depolarization. But in almost all cases, the amplitude of this EPSP is far above the magical point of threshold. So at this site, we'll now generate the opening of voltage-gated sodium channels through the initial opening of those ligand-gated ionotropic channels. And you had a question.
Yeah. So what did you say opens the nicotinic ligand-gated channels again?
Acetylcholine.
Oh, okay. I thought you said nicotine cells thrown off.
Nicotine opened the nicotinic acetylcholine receptors when Langley was walking along the muscle fiber. Okay. But of course, we don't use nicotine as a neurotransmitter, we use acetylcholine. So the nicotinic acetylcholine receptors will now bind the natural neurotransmitter acetylcholine to open them and let sodium come in. Yes? So the myoblasts during development form the muscle fibers, yes. So when you're working out at the gym, you're not generating new myoblasts. You're not increasing new muscle cells. You're increasing the diameter and size of already in place muscle fibers. So that means if we look at this thing, we see that this cell may be as much as a centimeter long. That means to have communication along this membrane, we need to invoke our old friend, the voltage-gated sodium channels, to propagate an action potential. If we were simply to use the nicotinic acetylcholine receptors and the response to acetylcholine, when the sodium would come in, the positive charge would flow out through leak channels all along the membrane, and this EPSP would decay within a matter of a few millimeters at most. So to have this function, we need to amplify the EPSP. And of course, the amplification of the ESP is carried out by the voltage-gated sodium channels that initiate and propagate the action potential. So it's exactly the same pattern. we release neurotransmitter, it binds to ligand-gated inotropic channels. When they open, sodium comes in, we initiate an action potential now that propagates along the membrane. And that's the electrical signaling event, which I now want to take into the movement of ions out of the sarcoplasmic reticulum. Yes?
What's our deactivator for the neurotransmitter in this case?
Oh, I should have mentioned that. Good point. Here in the cleft is something known as acetylcholinesterase. And the acetylcholinesterase converts acetylcholine into a molecule of acetate and a molecule of choline, neither of which are capable of opening the nicotinic acetylcholine receptor. So we're inactivating the acetylcholine. And what nerve agents do is they inactivate the acetylcholinesterase. So then we get a buildup of acetylcholine in the synaptic cleft. And that leads first to overactivity, and subsequently, because of desensitization of the acetylcholine receptors, we basically get no activity. Yes?
The acetylcholine is in the synapse only, right? Only the sodium comes into the cell.
So the question here is a really important one. The acetylcholine does not enter the postsynaptic cell. Only the ions enter the postsynaptic cell, and that's true for all of these neurotransmitters. They themselves don't go across the phospholipid bilayer. They're too big and they're too charged. So their communication is through the opening of a ligand-gated ionotropic channel. I gotta get going. What is it?
Fibers behave like an axon, basically.
So the fibers behave like an axon, yes. So we have voltage-gated sodium channels which propagate the potential all along the membrane. So now what I want to do is I want to elaborate that structure in this upper panel to describe the organization of the membrane systems in the muscle fiber. So we'll take this panel and label it as muscle cell, we'll label it as muscle cell membrane systems. Muscle fiber membrane systems. So this structure is again going to be a representation of the muscle cell. This is the plasma membrane. In muscle cells, the plasma membrane has a name. It's called the sarcolemma. Now, if we were to stand on this muscle fiber and take a walk, we would see that we wouldn't walk very far until we'd find this great pit. And every micrometer or so, we would see another one of these pits. We could rappel down the side of the pit, walk across the bottom, and climb out the other side. What I'm trying to say here is these structures known as transverse tubules, that the membrane, the phospholipid bilayer of the T tubule, is an extension of the plasma membrane. From the upper panel, we can imagine the site of the neuromuscular synapse. We can now imagine the postsynaptic membrane. And I won't go through all the details, but the excitatory postsynaptic potential is generating enough positive charge that it's now going to lead to the opening of these voltage-gated sodium channels. Now, those voltage-gated sodium channels are not confined just to the surface sarcolemma. Those voltage-gated sodium channels extend down and are included within the T-tubule membrane. So what do we have so far? We have the sarcolemma and an extension of the sarcolemma down to form the T-tubule membranes. And if we look carefully here, and I'll go over here. If we look carefully, we see that there's an extension of the T-tubule membrane. And then here, right next door, is a membrane of the sarcoplasmic reticulum. The sarcoplasmic reticulum, the SR, is a membrane-bound bag of calcium. So these two membranes, the red membrane of the sarcolemma and the blue membrane of the sarcoplasmic reticulum, these are two separate and distinct membranes. And what we see is that in the sarcoplasmic reticulum, there are proteins known as dihydropyridine receptors. These dihydropyridine receptors we abbreviate as DHP receptors. Now, the dihydropyridine receptors are transmembrane protein molecules, and they extend across the T-tubule membrane. For each of these dihydropyridine receptors, there is a corresponding transmembrane protein in the sarcoplasmic reticulum. The corresponding protein here in the sarcoplasmic reticulum, these proteins are said to be ryanodine receptors. Known as RYR receptors. Now, these dihydropyridine receptors, have this extension, a cytoplasmic extension, shown here as sort of being a little outcropping. That particular outcropping now is organized in such a way that it fits down in the channel of the ryanodine receptor. It actually plugs the channel. But this extension is also a voltage sensor. Now, it's not like the S4 region, but it does function as a voltage sensor. Now, watch what happens here. We've said that the voltage-gated sodium channels are on the sarcolemma proper, but they also extend down in the T-tubule membrane. So when the action potential is initiated by the release of neurotransmitter, the opening of those ligand-gated channels, and then the opening of the voltage-gated sodium channels, they propagate along the plasma membrane and down the T-tubule membrane. The opening of those voltage-gated sodium channels now delivers positive charge into the narrow gap between the T-tubule membrane and the SR membrane, known as the T-SR junction. We said that this sort of tail on the dihydropyridine receptor is a voltage sensor. When it sees positive charge, it moves. And when it moves, when it moves, it unplugs the ryanodine receptor. Because the ryanodine receptor is a calcium channel, calcium can now pour out of the sarcoplasmic reticulum, because the calcium concentration here is in the neighborhood of 1 to 2 millimolar. But out here in the cytoplasm, it's in the neighborhood of 0.1 micromolar. So it's a tremendous gradient. So when you pull the plug out of the bottle, the calcium pours out of the sarcoplasmic reticulum into the cytoplasm. So what have we done so far? What we have done is we have released neurotransmitter to bind to ligand-gated ionotropic channels, to generate an excitatory postsynaptic potential, to trigger an action potential which propagates along the plasma membrane and down the T-tubule system. The positive charge delivered by the action potential causes this tail, this nipple, if you will, in the ryanodine receptor to be pulled out of the ryanodine receptor and allow for calcium now to be released from the membrane-bound bag and into the cytoplasm. So any questions up to there? Yes?
Which one's the voltage sensor?
So which one is the voltage sensor? It's the dihydropyridine receptor here is the voltage sensor. It's only a section. I don't remember which loop it is. It's one of the cytoplasmic loops that's now the voltage sensor. Okay, well, let's take this idea now and superimpose what we're talking about here onto the cytoskeletal organization of the muscle fiber. So what we're going to do now is we're going to superimpose the story of the release of calcium from internal stores onto the description of the cytoskeletal organization. And we'll take this panel and label it as the cytoskeletal organization of the skeletal muscle cell. So we'll label this as the cytoskeletal try again. The cytoskeletal organization of a skeletal. You can read voluntary muscle cell. Now the players here are going to be first something known as alpha-actinin. Alpha-actinin forms Z-lines. The second character is going to be myosin, thick filaments. The myosin thick filaments are made of bundles of myosin. The third character is going to be the actin. Let me just call it this, the thin filament. And the thin filament is composed of filamentous actin, troponin C, or troponin complex, which is the calcium binding protein, And third is a molecule called tropomyosin. All of these taken together are organized into a structure called the sarcomere. So let's put those into place. So again, this is the muscle fiber. Then attached to the cytoplasmic side of the plasma membrane are going to be these zigzag structures known as Z-lines. They were described as Z-lines entirely upon their morphology. We now know that they're composed of an actin binding protein called alpha-actinin. These Z-lines occur at regular intervals, at an interval of about 1 to 2 micrometers. Attached to the Z-lines, I'm going to put just a blue line in here. Attached to the Z-line will be these blue parallel lines, which I'm now going to label as the actin thin filament. If we look at one of these actin thin filaments, the actin thin filaments, as I've mentioned here, are composed of 3 separate proteins. So each actin filament is composed of a polymer of G-actins. So this really is nothing more than what we referred to earlier as F-actin. Then there is this ribbon-like molecule called tropomyosin. The ribbon-like molecule extends over 6 to 7 of these individual actin monomers. So this purple structure is meant to represent the tropomyosin. The third character in this story will be the troponin complex, and each one of these tropomyosin molecules has attached to it, I'll show it in black, a troponin complex. Now, there are 3 molecules in the troponin complex. The one I want you to hold on to is the one called troponin C. Troponin C binds calcium. If we now drill down and look underneath the tropomyosin molecule, if we sort of lift up the tropomyosin molecule, we find on each one of the actins, I'm going to depict it as a red dot, on each one of the actins, there is a myosin binding site. Now let's go back up here, and I want to put the myosin bundle in. The myosin bundle is shown here with the myosin heads reaching out. So this is the myosin thick filament. So, envision this as having these myosin heads that we talked about before, reaching up and trying to contact the actin thin filaments. So imagine these myosin heads trying to reach up and contact the actin thin filaments. I've shown it in 2 dimensions, but there would actually be 6 of these around the circle. And each of these myosin heads is trying to reach up and contact the actin thin filament, is trying to bind to this red dot here. But under normal resting conditions, it reaches up, and it can't get to that binding site because the tropomyosin's in the way. But now, when the action potential arrives at the TSR junction, and that action potential then causes the tail on the dihydropyridine receptor to be pulled out of the ryanodine receptor, and calcium flows out. So now calcium, which has been released from the sarcoplasmic reticulum, that calcium now can bind to this molecule of troponin. When the molecule of calcium binds the troponin, the tropomyosin moves out of the way. And when the tropomyosin moves out of the way, it exposes the myosin binding site on the actin. So another way of thinking about this is that if you imagine my fist here trying to reach up and contact one of those those myosin binding sites, it can't get to there because this ribbon-like molecule is in the way. But when calcium binds to the troponin C, it changes the orientation of that tropomyosin so that now it turns on edge. So it goes from this flat ribbon-like structure, which was occluding the binding site for the myosin, and moves it out of the way. The myosin head now can reach up and snap onto that myosin binding site, attach to the myosin binding site. So what I'm going to do in this back board is describe actomyosin bridge formation. And the power stroke. So here's the actin monomer. The myosin head looks like this. Now remember, we said an important aspect of the myosin head early on. We described this region of the myosin head as having an ATP binding site. What's more is the head is an ATPase. So it converts ATP to inorganic phosphate plus ADP. So now this site contains ADP and inorganic phosphate. Then there's the red dot. When the tropomyosin's moved out of the way, which we showed above, when the tropomyosin folds down into the groove and moves out of the way, that provides an open pathway for this myosin head to come up and latch on to this site. When the site is now bound to the actin, at that point now, the ADP and PI leave. When the ADP and PI leave, this leads to a change in the configuration of the myosin head. So that where this myosin head at this position is pointed more or less straight up, it's going to now bend over. So it now bends over into this new position. And when it bends over, when it goes from this upright position to bent over, it tugs on the actin thin filaments. So now if we go to the upper panel, again here, when these myosin heads reach up and contact the actin, they're pulling on the actin thin filaments and they're shortening the sarcomere. By pulling on the actin thin filaments, they'll shorten the sarcomere by 20%. So it'll go from about 1 millimeter to about 0.8 millimeters. All of these are laid out in series. So that means that when we shorten these sarcomeres by 20%, we take the muscle fiber, pick a number, 1 millimeter, and make it now 0.8 millimeters. So we shorten the whole muscle. And that's accomplished by these myosin heads reaching up, contacting the actin, and changing their configuration, tugging the Z lines together. To finish this, the question is, how do we end this? How do we stop it? The first thing we have to have is that adenosine triphosphate now binds back again to this bent over site. Because remember, the ADP MPI is left. So now ATP goes in here. That tells this myosin head to let go of the actin thin filament and returns to its upright position. So it basically gets recocked. In the process of recocking, the ATP is converted to ADP MPI. So now the ADP NPI is in position such that when the myosin head binds the actin, the ADP NPI leave, and we get another power strip. This continues as long as two situations are met. As long as there's ATP to release the actomyosin bridge, and as long as there's calcium to bind to troponin C. So the way we end this is we now use pumps on the sarcoplasmic reticulum. So now we use pumps on the sarcoplasmic reticulum to return calcium back to the SR. As the calcium concentrations of the cytoplasm begin to drop, they now release their hold on the troponin C. The tropomyocytes move back to their old positions, and the myosin heads now are occluded from binding to the actin. And that's the way contraction comes to an end, and we can leave it there.